everyone. My name is Kathleen Nolan, and I'm here to tell you about my master's research titled Blooming Barcodes, DNA Barcoding for Freshwater Microalgae Identification. This project is taking place in the College of Biological Science in the Department of Integrative Biology, under the co-advisory of Drs. Andreas Hayland and Robert Hanner. I'm excited to present my research to you today, and thanks in advance for your attention. I hope you enjoy. Okay, so before we get into the meat and potatoes of my presentation, I wanted to take a moment to discuss microalgae. What are they and why are they important? Microalgae are single-celled photosynthetic organisms that are abundant in aquatic ecosystems. They are an incredibly diverse group of organisms with representatives across the tree of life. They play important roles both in aquatic ecosystem functioning and in the biotechnology sector. The biotechnological value of microalgae is astounding, and they have applications in several industries, such as nutraceuticals, pharmaceuticals, bioplastics, biofuels, agriculture, and aquaculture. They're beneficial in ecosystems because they form the basis of aquatic food webs, as we can see in this video of a tardigrade munching on some microalgae. However, when any one species grows out of control in an ecosystem, we can see harmful algae blooms, or HABs, which have tons of negative ecological consequences, like the creation of anoxic zones and fish die-offs. Other negative consequences of dense microalgal growth include toxin accumulation and biofouling of membranes in water treatment facilities, both of which are costly and time-consuming to fix. Given the importance of microalgae in the environment and in applied systems, it's important for researchers to be able to identify them quickly and accurately. So how do we identify microalgae? Well, microalgae have historically been identified by microscopy, that is, by observing the algae's morphological traits under a microscope. This method is fascinating and beautiful. Green microalgae, seen to the right, and cyanobacteria along the bottom of this slide can form complex colonies with beautiful patterns. And diatoms have intricate shells made out of glass that form complex arrangements. Again, microalgae are incredibly diverse, and this poses challenges for their identification due to the sheer number of species, many of which are cryptic or underrepresented in the literature. Furthermore, many species share morphological traits, making them difficult to distinguish from one another. Microscopy is a well-established method, but it requires lots of time and morphological expertise to complete accurately. An appealing alternative to traditional microscopy lies in the realm of molecular identification. One such method, known as DNA barcoding, involves the isolation and amplification of a single fragment of DNA from an individual target organism, which can then be sequenced and matched against a molecular database to obtain a species identification. This is becoming increasingly popular as high-throughput sequencing gets better and cheaper, and we expect to see this method increase in popularity in upcoming years. However, this method does have some drawbacks. First, it relies on well-populated molecular reference libraries, such as NCBI's GenBank, to match the DNA barcodes against. In the specific case of microalgae, these reference libraries are quite underdeveloped. Secondly, several methodological gaps exist in the DNA barcoding of microalgae, such as the lack of optimized reaction conditions and the lack of standardization in the analytical process for DNA barcode data. These gaps must be addressed before this methodology can be effectively implemented in the identification of microalgae. Another promising molecular identification tool is known as DNA metabarcoding. DNA metabarcoding relies on the same fundamental principles as DNA barcoding, but instead of identifying a DNA sequence from a single organism, this method sequences DNA from any and all organisms present in the sample. This allows us to obtain a whole community snapshot of a community, which is very valuable when asking ecological questions. By applying DNA barcoding methods to heterogeneous or mixed samples, we can gain lots of new information and insight about a community. However, the same drawbacks exist with this method as with DNA barcoding, as this method also relies on those well-populated reference databases. So given these identification tools, I wanted to address the following objectives with my research. First, I want to contribute to reference library building efforts by generating several high-quality DNA barcodes from microalgae strains. I intend to barcode strains that have been professionally identified and cultured by algal facilities. Then I will move into isolating monocultures from the environment using an ultra-dilution assay. I hope to use this library building activity to supplement the data already available for DNA barcoding of microalgae, and to compile a high-quality barcode reference data set with existing data plus data I collected, which can be used for future barcoding. I then want to use this reference data set to metabarcode mixed environmental samples. The results of this metabarcoding work will then be applied to several ecological models, which will allow me to interpret patterns in microalgal biodiversity. These ecological models will use environmental parameters like pH, water temperature, and concentration of nutrients to explain and compare different aspects of microalgal biodiversity across habitat types. So this research involves barcoding strains, that is, cultures with only one kind of microalgae. In order to obtain a microalgae strain, we can either purchase it from an algae facility or isolate it from the environment. I intend to do both, but on this slide, I want to talk about the methods I will use to isolate microalgae strains from environmental samples. This ultra-dilution-based method is called SHAPE, which stands for Single Cell High Throughput Assessment of Phytoplankton from the Environment. In order to isolate cells, I first count the cells in the sample under a microscope to determine the initial culture density. Then I dilute the culture to 1 cell per 20 microliters. Next, I plate the diluted mixture in 20 microliter increments into a 384 well microplate and count the number of cells in each well immediately after plating. If the well has only 1 cell upon plating, shown on this plate template in green, then that well is subsequently monitored and used to inoculate a new monoculture, which can then be barcoded. So as I mentioned before, I hope to DNA barcode type strains from algae facilities and monocultures from the environment. One of the major methodological gaps in microalgae barcoding lies in the choice of which DNA region to use. Several potential gene regions in the mitochondria, chloroplast, and ribosomal DNA of microalgae have been proposed as feasible barcodes, but they each have their associated pros and cons. In order to address this methodological gap, I'm going to try sequencing 8 different regions as barcode markers to see which one is the best. So far, I have tried amplifying the mitochondrial gene region ITS, the chloroplast gene regions TAF-A and RBCL, and the 23S ribosomal RNA gene region. The only region I've been able to barcode with so far is 23S rDNA, which is showing great promise for future work and will be discussed a little more on future slides. Once my barcoding endeavors are well underway, I hope to move into metabarcoding. I will collect filtered water samples from several local water bodies, which will allow me to sequence the selected barcode marker from all organisms present in the water sample. Then I will bioinformatically analyze this data to obtain a list of species and their relative DNA concentrations in the sample, which can act as a sort of proxy for organism abundance. Once I have these data, I intend to incorporate them into ecological models, which will allow me to look for interesting patterns in the microalgae community structures across many habitat types. So far, I've collected 28 separate live samples of microalgae, which has allowed me to practice and fine-tune the SHAPE ultra-dilution methodology. To date, I've successfully used SHAPE to isolate more than 20 monocultures from the environment. As I mentioned before, I was able to successfully generate barcodes for microalgae using the 23S ribosomal DNA gene, shown on the electrophoresis gel here. I obtained these barcodes from representative strains of the green algae, the cyanobacteria, and the diatoms. Specifically, I barcoded 5 strains, 4 of which were obtained from a professional culture collection, and 1 was obtained using SHAPE on samples from the Ottawa River. I'm still in the process of bioinformatically analyzing these barcodes, but the sequence data looks promising. In order to obtain these barcodes, I had to play around with the reaction conditions, and I'll continue to optimize the reactions for microalgae barcoding throughout my research. So given my preliminary results, I hope to continue to fine-tune the library building protocol for increased efficiency and accuracy. Once I have a reasonable reference library, I hope to move into meta-barcoding of environmental samples and eventually into building ecological models to compare microalgal communities across different ecosystems. Though my results are preliminary so far, I believe that my work will open new doors for folks interested in identifying microalgae, and I'm excited to continue contributing to this interesting cause. So to reiterate, this research is important because it will have broad implications for ecological monitoring of microalgae. This will improve our ability to detect and prevent HABs and to answer algae-related questions. Furthermore, this research will benefit the biotechnological sector because it will lead to improvements in microalgae characterization, thus increasing the efficiency with which we can bioprospect for valuable microalgae. Finally, this research will play an important role as a contribution to the global collection of microalgal barcodes, which will have far-reaching effects in future biodiversity monitoring work. By improving our capacity for microalgal identification, we can address all of these problems and more, ultimately improving the condition of aquatic ecosystems and those who rely on them. That concludes my presentation. Thank you so much for listening, and I hope you enjoyed. I'm happy to take any questions you may have at this time. Thank you.